Hello students, welcome to Smart IQ online classes, and I am your biology trainer Rajita, and today we are going to look into the topic biotechnology principles and processes. Now let's have a quick theory rundown of the topic so you may find it easier after watching the video. So let's get started. Now, what is biotechnology? The term biotechnology, as it is self-explanatory, includes both biology plus technology. Now, we are using technology to manipulate the biology of organisms in order to make it helpful for our use, okay, for industrial applications. And it was coined by a Hungarian engineer, Carl Erecki, in 1917 to describe a process of large-scale production of pigs. So the definition of biotechnology that I had told you before is slightly different from what they had thought earlier. Biotechnology was very specific to large-scale production of pigs back in the day in 1917 when it was first coined. But recently, the definition of biotechnology has been altered. It has been given by the European Federation of Biotechnology, or EFB. Okay, so according to this, biotechnology is the integrated use. Now, what is integrated? Integrated means it is an amalgamation. Okay, meaning it is a combination of biochemistry, microbiology and engineering sciences. Okay, so biochemistry and microbiology are life sciences and engineering sciences, okay, in order to achieve industrial application. Okay, so when I say biochemistry and microbiology, it is the life science part. It deals with the genome of the organisms, or how you manipulate the genome of the organisms in the first place. So in order for you to manipulate the genome of the organisms, you will need the help of engineering sciences. When I say engineering sciences, I specifically mean softwares that are designed for this purpose alone, that is for you to collect biological data, that is study the genome of the organism and the gene of interest, okay, and also to give you a analysis of what may happen if you were to manipulate a genome, or what can you manipulate the gene or the genome in the first place. So the engineering sciences, or the softwares in this part of the biotechnology, will help you understand how to effectively manipulate an organism, okay, in order to achieve industrial application. So why are we interested in manipulating the genomes of organisms in the first place? It is because we need to utilize it for our own application, okay, for industrial application. Specifically, when I say industrial application, it means large scale production of, say, some organic compounds, some hormones. As you all know, we've all been very familiar with the advent of biotechnology, and we are all using those products. So in order to achieve that, we need the integration of these three sciences, okay, and it is also to manipulate the capabilities of microorganisms, or cultured tissues or cells and parts thereof. Now, there are two, they're not divisions of biotechnology, but there is a time when biotechnology changed, and there was a time when biotechnology was something else. Not really something else, it was not as advanced as it is today. So according to the time scale, there is going to be old biotechnology, or the traditional biotechnology, and the modern biotechnologies that exist. Now, what is old biotechnology or the traditional biotechnology? Microorganisms were first used to produce organic compounds. Okay, they were simple organic compounds that the microorganisms were known to be using, okay, known to be producing. Okay, so citric acid was one among them. So there was a large scale breeding of microorganisms in order to produce some compounds like the citric acid and sometimes even formic acid, etc. They were also used for production of antibiotics like penicillin. So penicillin was the only effective antibiotic known back then, but now we have other synthetic antibiotics as well that are much more effective than penicillin. So penicillin was one of the very few antibiotics that were produced using old biotechnology. But here it is very important for you to remember the fact that in these processes only the natural capabilities of organisms are exploited. So what do I mean by natural capabilities? Now there is a strain of fungus that is known to produce penicillin. Okay, so we are only going to culture that, we are only going to grow that in order to get penicillin. Okay, we are not genetically manipulating one strain of a different fungus to give you penicillin. No, that is not happening. Okay, we know that a particular strain of the fungus causes the production of penicillin, so we are going to use only that. So we are not exploiting or manipulating the genome of organisms in any way. Whereas in modern biotechnology, it is a slightly different scenario. Now, take the case of human insulin. Now, as I told you, one of the products of the most popular products of the modern biotechnology applications is the production of human insulin, the large scale production of human insulin. Now, why is human insulin such an important part? Okay, because there are billions of people around the world who are suffering from the diabetes, insulin dependent diabetes mellitus and all that, and millions alone are millions are in India alone. So you do I hope you do realize how important it is for the production of insulin hormone in large scale or industrial scales. Okay, so human insulin production is a product of transgenic. Now, transgenic, what is a transgene? Transgene is a combination. Okay,